So the first laureate to speak today is Mary Claire King. All of you know her. Again, she has an unending list of prizes, awards, and nominations. And again, I would like to take you many years ago to one of the major contributions of Mary Claire, where many years ago, working with Alan Wilson, they were able to show that the difference between the human and mouse genome and ape genome, chimpanzee, is actually very small. Actually, they were able to show, mainly looking at structure and mobility and feature of proteins, also some DNA reannealing experiments, that we are very, very similar to chimpanzees. And they came with major insight that I think revolutionized the field. They suggested that you can't explain the difference in body structure and the way we think and act between us and chimpanzees just by looking at the sequence of coding regions of proteins. So they suggested that it has to do with regulation. Alteration in regulatory sequences need to be at the base of the difference between the species. The second major contribution of Mary Claire King that you are all aware of is doing at that time, almost mission impossible. You know that breast cancer is a very common disease. 1 out of 9 women are affected. The majority of cases are not due to inherited factors. There are a lot of environmental factors, and yet they are clearly familial cases. So Mary Claire King managed, using very advanced genetic methodologies at that time, advanced linkage analysis, and the analysis of families with early onset of breast cancer, and also based on their graduate student as a background in mathematics, she managed to locate what we call today the BRCA1. Actually, she gave this name to this gene on chromosome number 17. So she was, as I mentioned, was able to answer a very basic question in evolution. She demonstrated for the first time that predisposition not only to breast, but breast and ovarian and maybe other tumors by the identification and localization of BRCA1. And actually, this was a landmark study in the genetics of complex diseases. Mary also made major contributions in the study of other genetic diseases. Just to mention hearing loss and schizophrenia. By the way, her studies regarding hearing loss are in close collaboration with Karen Avraham, who is here with us at the Tel Aviv University. Then Mary Claire also developed a methodology for sequencing of mitochondrial DNA in human remnants. As a model human rights activist, she applied this methodology of mitochondrial DNA sequencing to the identification of kidnapped children in Argentina and to the identification of genocide victims in Rwanda, Bosnia, and Ethiopia. Please welcome Mary Claire King.
Many thanks. It's wonderful to be back at Tel Aviv University. Because you've heard a little bit about the story of my PhD thesis, I will tell you 2 points about it. The first is that as I was completing it, this must have been about 1972 or 3, Alan Wilson, my advisor, was here. He was at the Weizmann on sabbatical. And of course, in 1972 and 3, we did not have any rapid way of communicating. So every Friday afternoon, when people at the Weizmann were home cooking, Alan and I would speak by phone, and it cost a bloody fortune. And so we had to be very efficient, and I had to do that project with no wasted words. And then when Alan came back, we wrote it up. And Alan died of leukemia when he was only 58, and I miss him every day. I want to tell you one story about that project. I hadn't thought to tell it, but you'll like it. When my daughter Emily, who's now, of course, a middle-aged person, when she was 18, just finishing high school, she asked if we could go to Ireland. Part of our ancestry is Irish. So I said, sure. And at this time, Ireland still did not have wide highways. It had small roads, and one went from town to town on small roads. So I rented a car. It was a manual transmission car. I had not driven a manual transmission car for years. And of course, then and probably still, the Irish drive on the opposite side of the road from Americans and here. So I was focusing very carefully on the road, on driving on the proper side of the road and with this manual transmission. And I wasn't watching the sides at all. I was simply focused on the road. I realize this is not an Israeli thing to do, but I'm Irish, what can I say? So, but we were driving along, and suddenly Emily said, "Mommy, look!" So I stopped. And she pointed to a small roads, a small billboard beside the road. And this billboard was dark green, and it said, in its entirety, "Scientists have shown humans and chimpanzees are 99% the same. Drink Guinness." And Emily said, "Mommy, that's you." And I do not need to tell any of you who are parents of teenagers, you treasure these moments. 25 years later, I still remember it. Right? So my assignment today is to bring you up to date on the most recent results in our studies of inherited breast and ovarian cancer. I'm going to expand them a bit also to include advanced prostate cancer, as you shall hear. And in particular, how these recent results have led us to be able to claim now that we are moving from gene discovery to precision medicine and to public health. I want to start with one of my favorite people, Paul Brca. Paul Brca was working as a surgeon and a pathologist and a neurobiologist in France in the 19th century. This is the Brca for whom Broca's brain is named. But he was active in a wide variety of what we would now call biomedical research. And among his work was a treatise written in 1864 in Paris, the Traité de Tumeur, that described 4 large kindreds for whom he had been the surgeon of the patients with breast cancer in the youngest generation. And all I've done is convert his paragraphs into modern pedigree format. So what Dr. Brca was able to do was to obtain autopsy material from a number of the persons, the women who had died of breast cancer in each of the families, and to evaluate that autopsy material. And you can see, thank you so much, you can see that he describes in this family cancer of the breast that appears in multiple generations. And he also describes, presciently, I think, cancer, let me see if I can, may I use this as a pointer? Let me see if it works. Yes, perfect. Cancer in the abdomen, and he has a couple of cases of cancer in the liver, here, somewhere else. And he says in the traité, "I am finding in this autopsy material cancer in these organs, but," he says, "I think this is not cancer of these organs. I wonder," he says, "if these abdominal cancers may have invaded from the ovary." So I think that almost certainly what Dr. Brca was describing were early cases of breast and ovarian cancer in the same family. My friend Dominique Stoppa-Lyonnette at the Curie has tried to trace the families of Dr. Brca in France to see if we can find them and identify what their mutations in BRCA1 or BRCA2 might be. Thus far, she has not succeeded, and we wonder if perhaps those branches of the families eventually died out, but she continues to look. I like to try to imagine what might have happened if Dr. Brca, working in Paris in the 1860s, had been able to communicate with Mendel, working in Brno at the same time, and with Darwin, working in southern England at the same time. Suppose they had had Skype. If they had, they might well have saved us the intervening 130 years of slogging through this field, because they could have applied their minds to it very successfully. We know that Brca was a founding member of a society of free thinkers in Paris, and that he had read the work of Darwin, and indeed had written some letters to friends about it. We also know from recent discoveries in the library in Brno of Mendel that Mendel had read the work of Darwin and had made notes. There's notes in Origin, in Mendel's handwriting, comments on Darwin's work. But insofar as we know, none of them ever communicated with each other. I'm going to do one thing to my computer so I can see my own screen better. Please hold on just for a second. This is just me. Great. Concession to my eyes. So we're going to skip the next 130 years, and in so doing, we are going to skip the entire process of positional cloning. Those of you who are older than age 25 will understand exactly why I choose to skip this. Those of you who are less than age 25 will be blessed in never having to go through this process. The human genome, the fact of the human genome, not to mention all of the other genomes that have been sequenced, have saved us all from ever having to carry out positional cloning again. Suffice it to say that this process was the process used by a number of groups in a competition that went from roughly late 1990, early 1991, until 1994. There were probably 100 scientists, geneticists, all of us professionally trained, all of us working very hard, and probably 15 major groups in academia and in public sector, in government labs, and private labs, competing to clone BRCA1. And I was not the winner of that competition. It was cloned by a private company, but the process that was used was this positional cloning process, which was tedious, but did in fact require that one really think carefully about biology as one went along. We had to sequence de novo, from scratch, each time we had a new bit of chromosomal region in the area that we cared about. So we will skip all of that. And let me talk to you for a few minutes just with a couple of examples of families with whom we've worked for decades now, to give you a feel for what the mutations look like in BRCA1 and BRCA2, and what the clinical profiles of these families look like. BRCA1, as you've been told, was a gene that I had the pleasure of naming, and officially, of course, it stands for Berkeley, California. It stands for breast cancer, but unofficially, I've stepped on my punchline. Unofficially, it stands for Berkeley, California. This is family number 1, and family number 1 means that it was the first family that we identified. The family is English ancestry, lives in the Midwestern part of the States. And as you see, in this family, some of the principles elucidated by Dr. Brca obtain. That is, breast cancer is found in multiple generations. But one principle in this family, or one fact of this family that Brca did not describe in any of the families that he had treated, is that in this case we see clearly that predisposition to breast cancer can be inherited through either fathers or mothers, as here. BRCA1 is on chromosome 17, it's autosomal. Men have chromosome 17 also. They can have mutations in BRCA1 and are virtually always unaffected, not without exception, as we shall see, but virtually always. That means that mutations in the gene the phenotypes associated with mutations in the gene can skip generations simply because they are passed through fathers. It's still the case now, 20-some years after the cloning of BRCA1, that one of my great challenges in talking to medical students is to try to embed in their thinking that in discussing with a breast cancer patient or an ovarian cancer patient her family history, the physician must think both about the maternal side of the family and the paternal side of the family. As we'll see, roughly 50% of women who have mutations in these genes have inherited the mutation from their father, the other 50% from their mother. And if the mutation is inherited from the father and the family is small, there may be no other cases of breast or ovarian cancer in the family. But more about that later. A couple of other points I'd like to make about family number one are that we see both breast and ovarian cancer in the family. There's a fallopian tube cancer here, there's an ovarian cancer here. We also see that the breast cancers in the family occur at very young ages. I've intentionally shown you an outlier first. The ages in this family are, as you see, extremely young at diagnosis. Another point is that there are multiple cases of breast cancer in this family amongst women who do not carry a mutation in BRCA1. Breast cancer is a common disease. There will be cases of breast cancer for reasons having nothing to do with inherited genetics, even in families with inherited genetics. And fourth point is that occasionally, not often, but occasionally, we encounter women who are older, who carry the same mutation that has led to cancer in their, in this case, much younger daughter, but they have escaped. I think those reasons are largely stochastic, largely chance. The risk, as we shall see, of developing breast cancer amongst women who carry mutations in BRCA1 by the age of this woman, the risk is about 80%. 80% is not 100%, and she's extremely fortunate. Naturally, we did a study of these ladies, these elderly ladies who had not developed breast cancer despite carrying an unquestionably damaging mutation in the gene. This particular lady, when I was talking with her about all the possible non-genetic risk factors that might have protected her, drew herself up to her full height, which was about this, and said, "My dear, don't you think that if I knew what had protected me from breast cancer, I would have told my daughter?" So fair enough. After decades now of trying to sort through what those reasons might be, I think in the great majority of cases, elderly women who have escaped breast cancer despite a devastating mutation in BRCA1 or BRCA2 are simply fortunate. The cancer occurs when, but only when, a second mutation occurs in the critical tissue, either the breast ductal epithelial cells, these are the same cells that are responsible for milk, or in the epithelial cells that cover the ovary like a delicate blanket. Absent that second mutation, a woman with a mutation in BRCA1 or BRCA2 leads a totally normal, completely healthy life. One will never encounter more productive, terrific women than women with mutations in BRCA1 or BRCA2, but they do have this horrific susceptibility to breast or ovarian cancer. I want to show you this mutation because unlike the previous mutation, indeed unlike the great majority of mutations in BRCA1 and BRCA2, this mutation is a missense. The previous mutation that I showed you was, of course, as you saw, a small frame shift, a deletion of two base pairs right in the middle of the gene, leading to a virtually immediate truncation of the coding sequence. This is a missense, and it's a missense of a very particular sort. It's a missense in the ring domain of BRCA1, shown here, and indeed it's a missense in one of these residues that is responsible for binding zinc. My friend Rachel Clevitt at the University of Washington, who's a very fine structural biologist, elucidated why these missense mutations in particular matter, because the great majority of amino acid changes in BRCA1 have no effect at all on protein function. They're perfectly neutral, as of course are most changes in most genes that don't stop its reading frame. What Rachel and her students showed was that BRCA1 makes a heterodimer with BARD1, its partner, and as you see, they are very similar in structure. This is the N-terminus of BRCA1, the N-terminus of BARD1. Each of them contains zinc binding motifs, and we've very recently shown that comparable mutations in BARD1 have the same effect as this mutation in BRCA1 that I'm showing you. And as you can see from the pedigree, this mutation is co-inherited with breast cancer. Why? Why does this particular change of an amino acid matter? The family, incidentally, in this particular case, again, an American family of Northern German ancestry. The reason that loss of the capacity to bind zinc in this structure matters is that such loss destabilizes this structure, and destabilization of this structure leads to loss of ubiquitination activity on the part of the complex that includes BRCA1 and BARD1. So unlike the best known function of this complex, about which more in just a moment, the best known function of this complex is to be critical to the repair of DNA using the most accurate pathway for repair of DNA. But this is a little sidebar in that functional story, namely that this beautiful structure acts as a ubiquitin ligase, and in particular has as a substrate the estrogen receptor. One of the great puzzles of the biology of BRCA1 and BRCA2 had been why the cancers that are caused when their function is completely lost are breast, ovary, and as we shall see to a lesser extent prostate, and to a still lesser extent pancreas. In other words, all cancers that are in organs that are associated with steroid function. And the reason, we now think, is that a second function of the complex is ubiquitination of estrogen receptor, and to a lesser extent the other steroid receptors, processing those receptors so that they are not overproduced in the cell. And that loss of function, loss of this particular function of BRCA1 or BARD1 leads to overproduction or over inclusion of estrogen in the cell, and therefore a greater chance of survival even of a mutant cell. The last bit of that is still speculative. We're still trying to pin it down, but I think that this class of mutations finally allows us to link the organs in which these cancers occur with the genes themselves. This had been a paradox previously because BRCA1, BRCA2, all of these genes are expressed in all organ types, in all cell types, and so it was paradoxical that only certain organs were affected, but I think we're beginning to get a grip on that. BRCA1 has a sister gene, a younger, shall we say, younger in discovery, but larger sister, BRCA2. BRCA1 and BRCA2 do not share any primary sequence, but they do form part of the same complex, which, as I mentioned, and we will find out more about, is involved in DNA repair, critical to DNA repair. I wanted to show you a couple of examples of families with mutations in BRCA2, just so you'll get a feel for the differences between the clinical consequences of these genes. Again, this is an outlier family, not with respect to the type of mutation. The type of mutation is absolutely conventional. It's a 4-base pair deletion leading to a frame shift and, of course, a stop. But observe the kinds of cancers that we see. In this family, there are several men who have developed breast cancer. There's nothing else unusual about these men. We've carried out studies of their steroid hormones, of the binding globulins that work with the steroid hormones. They are all fine. They are all chromosomally fine. No one is Klinefelters. No one is mosaic Klinefelters. All of them are fertile. I haven't put all the children and grandchildren on here, but everyone is fertile. It's simply that BRCA2 does predispose to breast cancer in men. There is something, this family clearly is an outlier when you see all the men who are affected, and there may be something about this particular mutation. I don't know. This family's American, live in the Bay Area, and are of Dutch ancestry, and the mutation is from northern Netherlands, from the Maastricht area. And I'd love to look at all families that carry this mutation, regardless of where they now live, and see if we see this really extraordinary high risk of breast cancer in men associated with this particular mutation. I can't resist showing you just because it's such an unusual phenomenon. Here's another unusual story. Again, a mutation in BRCA2, a different 4-base pair deletion. This one, I can't remember the origin of this one. I think it's Swedish. But look at the cancers in these men. These are not breast cancers. These are prostate cancers. This family was ascertained by us because of the breast cancer here and here, it fit our profile of families we wish to look at. But when we evaluated the BRCA1 and BRCA2 sequences, identified this mutation in BRCA2, as you see, the cancers that subsequently we identified amongst carriers of the mutation were disproportionately men with prostate cancer. And as we'll see in a few minutes, there's now been a systematic study of the risk of advanced prostate cancer associated with mutations in BRCA2. So that's an emerging story. A question that arose almost immediately with the cloning of BRCA1 and then a year later of BRCA2 in the mid-1990s were first, what proportion of women carry mutations in these genes? And second, what's the risk, the actual age-by-age risk associated with mutations in these genes? The answer to the first question depends entirely on the population. Every mutation has an origin in time and space. All mutations in BRCA1 and BRCA2 are young in an evolutionary sense. The oldest one is one of the mutations that is common in the European Jewish population and dates to sometime in between the first temple period and the second temple period. And we know that because of the populations that carry the mutations and the migrations of various populations of the Jewish people in and about that time, which are, of course, documented. So we can tie actual history to actual mutations. The other mutations are all younger. And most of them, I shouldn't generalize, many of them date from the time of rapid emergence of large towns in the medieval period. And the one I just showed you is of that sort. So we're still trying to get a bead on proportion of people who carry mutations in BRCA1 and BRCA2, which, as I indicated, depends on one's specific ancestry. So in a place like the US, where we're all mutts, the frequency is, I think, probably about 1%, in contrast to European Jewry, in which the frequency is very well known and is 2.5%. But to return to the second question, what's the risk associated with carrying a mutation in one of these genes? This was a very controversial point for several years. And the reason it was controversial is that in order to identify BRCA1 and subsequently the next year BRCA2, we all worked with families of exactly the sorts I've just shown you. Those families all came from that period of attempting to clone BRCA1, BRCA2. And so, of course, those families included multiple individuals with cancer. And therefore, tautologically, they had high risk, or they had high incidence. The question was, if one identifies carriers of these mutations from the general population, as opposed to specifically selecting families with many people who have developed cancer, what risk will one see? So Efrat Levy-Lahad and her colleagues, centered at Shada Zedek, carried out a project throughout the country here, ascertaining mutation carriers for BRCA1 and BRCA2 in what is basically a demonstration project for a population-based screening approach. And those of you who were involved in this project will remember it, I hope, with humor, and not with if you were one of the, shall we say, index people, not with too much chagrin. What Efrat did was identify 8,000 healthy men. Healthy men, self-identified as Ashkenazi Jewish ancestry, at least 35 years old. They were told that would they be willing to participate in a genetic study, but the specifics were not told them at first because the idea was not to select for men who either had or did not have particular family histories of any type. So 8,000 men, this being Israel, 8,000 men said yes right away. It was absolutely terrific. And these men gave a small blood sample, and the 3 mutations that are very common in the European Jewish population, 2 mutations of BRCA1 and 1 mutation of BRCA2, were genotyped in Efrat's lab. And as you would expect, because it was already well known, 2.5% of these men carried one of those 3 mutations. And then for those 2.5% of men, their pedigrees were taken in great detail. The study was explained to them in enormous detail. And the mutation was traced through their family to identify all of the women in the family who carried the index man's mutation. As time went on, it became clear that some of these men were related to each other fairly closely, so that the number of apparently independent families was reduced from roughly 250, 260 to about 175 independent large kindreds. So then what Efrat and her group did, after tracing all of the each mutation in each family, was to go back and see what had happened to the women in each family who carried the mutation of the family. So we now have a series of women spread across 175 different kindreds who have been determined to carry a mutation in BRCA1 or BRCA2, not because they themselves had cancer or because there was a family history of cancer, but simply because they were related to some nice guy who agreed to be an index person in the study. So what was the risk of developing breast or ovarian cancer among these women? And that was determinable from the health records in your HMOs and hospitals and from the Israel Cancer Registry. So we now have an unbiased answer to the question, and here it is. So the risk of developing breast or ovarian cancer, and these are combined, whichever happened first, for women who carried a mutation in BRCA1 or BRCA2, regardless of their family history, ignoring their family history, is shown here. And as you can see, it's extremely high for BRCA1 and still high, not quite as high for BRCA2. So this really absolutely nailed the answer to that question, what are the risks of breast and ovarian cancer associated with mutations in these genes? They are high. And it doesn't matter if one comes from a family with 7, 8, 10 breast cancer cases, or if one comes from a small family in which one is the only person with breast cancer, it's the mutation that matters. Now, this really nailed the theses to the door, as we Protestants like to say, but there was a non-trivial reason that it was a complex question. And the non-trivial reason is that there is another factor at play here in addition to genetics. So what Efrat and her colleagues did in order to get a handle on this second non-genetic factor was to take all of the women who are represented on this slide, who are mutation carriers ascertained through these healthy men, and to separate them by their year of birth. So the midpoint year of birth of all of the women in this project was 1958. Nothing magical about 1958, it's just that half the women were born before that and half were born after that. And if we look at the risks of breast or ovarian cancer to mutation carriers among women born in the earlier half of the cohort versus the later half of the cohort, here's what we see. To wit, age by age, the risk of developing breast or ovarian cancer is substantially higher for women who were born more recently than for women who were born earlier. Now, a little of this, maybe 2 or 3 years of this, is better diagnoses, is people having more mammograms, mammograms being better, and so on. But that will gain 1, 2, or 3 years. It doesn't remotely account for this difference. This result has been now replicated in multiple studies in multiple different populations with very different baseline risks. But the phenomenon has been shown over and over to obtain, namely that more recent year of birth, more recent birth cohorts are at higher risks given the same mutation, age by age, for particularly breast and to some extent ovarian cancer. The reason for this difference cannot be genetic. Right? These are the same women in the same families and the time is too short. The reason for the difference must be the non-genetic risk factors for breast cancer, which are responsible for the rapid increase in breast cancer having nothing to do with inherited genetics that obtains now worldwide. And those risk factors are early age at beginning to menstruate, so early age at puberty, which is associated with good diets in young children, and later age at first pregnancy, which is associated with education. So that interval between menarche, that is beginning of menstruation, and first pregnancy, the longer that interval, the higher the subsequent risk of breast cancer. And that's been shown in epidemiologic studies having nothing to do with genetics, going back now close to 50 years. Been shown worldwide and clearly obtains also among carriers of these mutations. This is not to say that we wish for women to go back to a time and environment in which breast cancer risks were lower. We are quite happy with being modern women, but it does mean that we have to think about this problem in a rational way, incorporating those non-genetic risk factors. So let me turn next to the question of how to address the reality of these risks. Shortly after BRCA1 and BRCA2 were cloned, a number of clinical collaborative groups working in North America, Europe, and here undertook to address the question of whether prophylactic, that is to say risk-reducing preventive surgery, would ameliorate the outcomes of risk in mutation carriers. And the first of these studies was carried out with a group centered at Mayo Clinic. And let me set up the design for you. As you, of course, know, Mayo Clinic in Rochester, Minnesota, has received for, oh gosh, over 100 years now, referrals for women in various sorts of women and men, in various sorts of tertiary situations, including having high risk, by virtue of their family history, of breast cancer. And over the decades, way before anybody had sorted out any inherited genetics of this trait, women who presented at Mayo Clinic with severe family history of breast cancer, despite being completely clear of symptoms themselves, had to confront the question of whether they wished to have preventative removal of the breast, that is prophylactic mastectomy, or not. And they were doing so on the basis of family history. That's all they had to go on. That's what there was. As you would expect, some women decided yes, some women decided no. Time went on, and over the decades it was possible to follow who developed breast cancer among women who had made one decision versus the other. And after BRCA1 and BRCA2 were cloned, it was possible to go back to the pathology specimens that had been collected for these women at the time of their initial intake, whether or not they'd had surgery, and to painstakingly in those days, because then it was painstaking, it's not anymore, sequence BRCA1 and BRCA2 and determine in retrospect who had mutations in these genes, even though neither they nor the surgeons knew it at the time. Does it make sense what I'm saying? Okay, terrific. So then what was done was to create the subset of women who had mutations in one of the genes and to ask what was the effect of the prophylactic mastectomy. And here's that story. So as we see, so everybody on this slide had a mutation in BRCA1 or BRCA2, although she did not know it when she made her decision. And as you see, among women who decided to have prophylactic bilateral mastectomy, or bilateral prophylactic mastectomy, breast cancer occurred, but in only 2 women out of something over 100 in this group. Among women who decided to undergo surveillance, and it was excellent surveillance, we're talking about Mayo Clinic follow-up, the risks over the intervening 30 years had been exactly what we would have expected based on Efrat's subsequent study. I, of course, being a skeptical person, said, why were there even 2 breast cancers after prophylactic mastectomy? And the reason was that some surgeons at that time, indeed I think still, left the nipple in place, which leaves some of the ducts that are vulnerable still in place. It's still, obviously the risk is much lower, but it wasn't 0, which it would have been otherwise. So this is the story of risk reduction in consequence of prophylactic bilateral mastectomy. A second question was, if we remove the ovaries and fallopian tubes of women who carry mutations in these genes, what do we do to risk? In this case, again, a large collaborative group of hospitals in primarily North America undertook to address the question and were able to do so in a prospective way. So as soon as BRCA1 and BRCA2 were cloned, women who learned through referrals to these tertiary and quaternary care facilities that they carried mutations in these genes were offered the option of prophylactic removal of the ovaries and fallopian tubes, assuming they were at least 40 years old or so at the time. And then they were followed prospectively to see what happened to them, and here's what happened. And the cancers that were followed in them were not only ovarian and fallopian tube cancer, but also breast cancer. So what we see here is that women who underwent prophylactic salpingo-oophorectomy, that means removal of the ovaries and fallopian tubes. These cancers, many of these cancers begin in the fallopian tubes and then spread to the ovary. By the time they are detected, they are already in the ovary. That those women had still a not 0 incidence of cancers. Most of these are breast cancers. I'll come back to that in just a second. But those who did not have the preventive surgery had a much more you don't need a statistic to tell you that there's a difference here, a much more severe risk, even over a period of just 6 years. So this led to, and I'll show you the actual script in a moment, this led to a recommendation by the International Oncologist Group that women who learn that they have a mutation in BRCA1 or BRCA2 have, when they have completed their families or at about age 40, preventive removal of the fallopian tube and ovary. Now, why does this help for breast cancer risk? It turns out that if one separates these risks into ovary and fallopian tube on the one hand and breast on the other, the reduction in breast cancer risk if one has that surgery by about age 40 is about 50%. So it's a dramatic reduction, still high, but it's a dramatic reduction for a surgery on a different organ. The reason, as you would expect, is that one has drastically reduced the endogenous estrogen level. Now, a woman who undergoes essentially surgical menopause at the age of 38 or 40, in the vast majority of cases, needs replacement estrogen. But the replacement estrogen that can be used can be titrated for the individual woman, and for the typical Western woman, because all of us have been fabulous estrogen-making machines until we entered menopause, for the typical Western woman, the amount of estrogen replacement that she requires for good quality of life is about a third of what she makes endogenously. So she is being exposed to much less estrogen than she would have been if she were still making her own estrogen through her ovaries. So this procedure has led, in many places, including here, I think, to a very strong preference for preventive removal of the ovaries and fallopian tubes, and then to consideration of when and if to schedule prophylactic mastectomy in addition. And often, in my experience, often a woman's decision about prophylactic mastectomy is made either when she has her first breast cancer diagnosed herself or if her mother died of breast cancer. That's not the most completely linear reason to make the decision, but I can certainly understand the perspective. Let me come now to the question of the relevance of BRCA1 and BRCA2 to treatment of breast and ovarian cancer. As I've alluded to a couple of times, BRCA1 and BRCA2 are part of a large complex of perhaps 40 proteins that are critical to the main pathway of DNA repair, and this is called the homologous recombination repair pathway. This pathway is responsible not only for DNA repair, but is also critical for stability of recombination. It's an absolutely central pathway to cell division and to just normal growth and development. And the way that this pathway works is shown here on the left. That is, we see in normal healthy cells that when a lesion occurs, which of course happens all the time, perfectly spontaneously in all cells in all people, this pathway goes into action. Repair, I can't point too well with this thing, but repair is made and the DNA goes on about its life. There is an alternate repair pathway which is both less productive, it works less well, and makes more errors, which is called the alternate enjoining pathway, and that's shown here, and that's also active. If we have a cell, let's say in the ovary, that has no BRCA1, say, as a result of combination of an inherited mutation on one copy of the BRCA1 sequence and an ovarian cell-specific mutation on the other copy, so now both copies have lost their function. So we have no BRCA1 function. The consequence of that is that this whole complex is now no longer functional. We have no homologous recombination repair when a mutation occurs in that ovarian cell, for example, a mutation in, say, P53, and the repair pathway is pushed into this alternate enjoining pathway, which is error-prone and makes mistakes and mutations accumulate. What a couple of groups of bright biochemists thought was, and these bright biochemists were on the one hand Thomas Halliday working in Stockholm and Alan Ashworth at the time working in London. Alan's now in San Francisco. What each of their groups thought was, knowing each of these pathways in considerable detail, and knowing in particular that this alternate enjoining pathway has as one of its principal components the PARP enzyme, if one could inhibit this pathway, then what would happen to cancer cells? They would not be able to repair DNA at all. They would not have that pathway functional, and they would not have this pathway functional. In other words, this pathway is already abrogated because of the mutations inherited and somatic that have occurred naturally, and we are about to medically kill off this pathway, which means that no repair can happen. What's that going to do to the effectiveness of conventional chemotherapy? It will make it much more effective, because conventional chemotherapy makes mutations in cells, and if the cells cannot repair those mutations, the cancers will be destroyed more effectively. And this turns out to work beautifully. Now, there were a few hiccups along the way. The first hiccup was that in some of the early clinical, so it worked beautifully in cells, it worked beautifully in mice, it worked beautifully in phase 1 trials, showed that there were not sufficient side effects to preclude its use in large randomized trials. Some of the first randomized trials had two problems. One problem was that the drug used did not in fact inhibit PARP, clearly an impediment. And the second problem was that the patients were not stratified by whether or not they had mutations in BRCA1 or BRCA2. I mean, as I've said and as we heard earlier, most breast cancer patients do not have inherited mutations in BRCA1 or BRCA2. So all that took a while to sort out. There was drama, you can only imagine. And then at the end of 2016, a trial was carried out that was well designed, well analyzed, and the results are shown here. And the results are basically triaged into, patients, in this case ovarian cancer patients, were upon recurrence following use of conventional good chemotherapy in the first round, were then randomized to take an inhibitor of PARP or not for their second round when they recurred. And the results have been effectively triaged into whether it was clear that the patient had an inherited mutation in BRCA1 or BRCA2, and in this top group, which are those patients, so the most crystal clear group, the improvement in median months to recurrence among those who were randomized to a PARP inhibitor was 21 months versus 5.5 months amongst those randomized to placebo. And the middle group are women for whom it was not clear that they had an inherited mutation in BRCA1 or BRCA2, but their cells clearly showed absence of activity of either BRCA1 or BRCA2. And as you see, again, a significant difference. And the third group are women in whom the cells, either there wasn't a cellular test done, or the cells did not indicate a loss of protein activity of BRCA1 or BRCA2. And there is still a difference here. I think there's several things happening in this group. I think, well, some of them weren't tested. The cells weren't tested. In some of them, I think what we have is epigenetic loss of BRCA1 or BRCA2. And a third thing that I think is happening here is that other genes that encode proteins that are critical to this pathway, about which we are about to hear more, were the ones that were mutant, and only BRCA1 and BRCA2 were tested. So this story has led to the introduction of PARP inhibitors much more broadly, and indeed to increasingly now to their use for breast cancer patients who have inherited mutations in these genes as well. I mentioned a few moments ago that our National Comprehensive Cancer Network of oncologists have made some guidelines about recommended action for women who learn that they have mutations in BRCA1 or BRCA2. Those are shown here, that women who have such mutations have annual screening by a modern technology from age 25. In the case, clearly, of families in which breast cancers have occurred as early as the 20s, we move that even younger. That they discuss the option of risk-reducing mastectomy, what's come to be known as the Angelina Jolie option. But then, and this is strong for a consensus group, that they actually be recommended to have risk-reducing removal of ovaries and fallopian tubes between the ages of 35 and 40 or upon completion of their families, with the understanding that for the great majority of women that will mean the use of replacement hormones until the normal age of menopause of about 50 or so. I've put in parentheses the result of an extremely nice study from Steve Nerod's group out of Toronto, published in 2014, There had been, as you would expect, there had been consideration of the possibility that these interventions might increase other forms of mortality, for example, heart disease. They do not. What Nerad and his group showed was that among women who had undertaken this procedure here, this oophorectomy, the reduction in all-cause mortality in consequence of the procedure, obviously primarily reduction in mortality from breast or ovarian cancer, but reduction in all-cause mortality was 77%. So we are not displacing breast and ovarian cancer with other causes of mortality. And then for treatment, that PARP inhibitor treatment be considered for BRCA1, originally BRCA null ovarian cancers, now breast cancers also. So what are we up against? Clearly, it is useful for women to know if they carry a mutation in one of these genes. Why isn't that obvious and offered to everyone all the time? Well, it's a very good question. There are some challenges. First, as I've already said, about half of patients with mutations in BRCA1 or BRCA2 are surprised to learn that because they have no close family history of breast or ovarian cancer. Second, there are thousands, an essentially unlimited number of different mutations in BRCA1 or BRCA2. At home, where I work with a population of very heterogeneous ancestries, and indeed in this country, where the country as a whole is highly heterogeneous genetically, if a woman presents, in Efrat's lab, day before yesterday, we were evaluating BRCA1 and BRCA2 sequences for women of non-European Jewish ancestry, so women of various Sephardi ancestries. And the mutations that we were seeing in BRCA1 or BRCA2 were the same classes that I've shown you. They were stops. They were mutations I'd never seen before. Efrat had never seen them before. I mean, new mutations are identified all the time if one begins to work with a population with which one has not worked recently, or because simply new mutations happen. Stuff happens. So in the early days, this was a terrible challenge because the sequencing was very slow and very expensive because it was slow. It was close to manual sequencing. Now, that's no longer an impediment. Next generation sequencing has come to the fore. Sequencing is extremely inexpensive and comprehensive, obviously if done properly. And the third challenge is that BRCA1 and BRCA2 are not the only genes involved. I've already told you this story, that half of patients who learn that they have a mutation in BRCA1 or BRCA2 are surprised because they have no close family history of breast or ovarian cancer. Here are two patients from one of our studies of American women of Jewish ancestry. And as you see, here are two women with mutations. We were surprised. They were surprised. What's up? Manishma, I would say, right? Here's what's up. The mutation in each case is inherited from the father, and the sisters were lucky and didn't inherit the mutation of the family. So it's a combination of small families, paternal inheritance, and good luck on the part of the other women in the family. The second impediment that I just mentioned was that there are thousands of different mutations in BRCA1 or BRCA2. Each vertical bar on each of these cartoons represents a different mutation. Some of them are extremely common in some populations, relatively speaking, of course, as the three mutations in persons of European Jewish ancestry. Others are private to one family. But this is all by way of saying that literally for any family, but relevantly for a family whose ancestral mutations one does not know, which is most families in this country, and certainly most families in my country, one really needs complete sequencing of both genes. Add to this, I won't show an example, but add to this the fact that some mutations are cryptic and one cannot detect them by conventional methods, and one must use next-generation sequencing methods because one must sequence all of the non-coding regions as well as the coding regions, and one has a technological challenge, now met, but it was a technological challenge for years. The third impediment that I mentioned is that BRCA1 and BRCA2 encode proteins that are part of this DNA repair complex, and several of the other proteins in this complex, if they undergo mutations analogous to those of BRCA1 or BRCA2, have the same consequence to breast cancer risk, ovarian cancer risk, or both, and those are shown here. Several years ago, when it became possible in a medium-sized lab like mine to carry out next-generation sequencing, we decided to tackle this problem by developing a gene panel, which we named for Brca. And that panel allowed us to sequence efficiently, inexpensively, and thoroughly all of the genes that could either were known to or could potentially be relevant to breast and ovarian cancer risk. And we introduced that. We did not patent it. It became, and the whole reason we did not patent it is we wanted it to be picked up by as many different entities as possible who would then buy the oligonucleotides that formed this panel from the company that made them. And we had nothing to do with the company, Agilent. They simply made the oligos. And then every place that decided to use this same approach would adjust it a little in the way that made sense for their particular needs. And that's exactly what happened. In consequence, the cost of sequencing, well, originally BRCA1 and BRCA2, and then subsequently all the genes, dropped from $3,800 per sample to about $200 per sample. This was enabled in my country because of the Supreme Court decision in 2013 that abrogated patents on genes. And because our methodology had been out there since 2010, it had been used in research laboratories, but could not be used clinically because of the patent and therefore the monopoly that was held by the company that had actually successfully been the first to clone BRCA1. Obviously not map it or identify it, but clone it. Because this technology was out there, literally overnight, between the time of the Supreme Court decision and the next morning, the capacity to sequence all genes for a much, much lower price came online. So it's a matter of technology being ready, but the fact of technology and policy converging, in this case, with a happy result. One consequence of having a panel that is completely thorough in targeting every base pair across all of the critical genes is that one picks up some of the cryptic mutations of the sorts that I show here. In addition to the classes of mutations that I showed you earlier, one picks up big deletions like this one, little deletions like this one that are cryptic, large duplications, enormous deletions, and so on. And one does so perfectly straightforwardly. So, of course, we have applied this technology to a number of very specific questions, and let me just give you some answers. One interesting question that I'm frequently asked by women of European Jewish ancestry is, if I do not carry a mutation that is one of the 3 known Jewish alleles, what's the chance that I carry some other mutation, either in BRCA1 or BRCA2, or in some other gene? Here's the answer. So among women whom we asked to join our study, because they had been identified, because they'd been diagnosed with invasive breast cancer, regardless of their age at diagnosis, regardless of their family history, among those women, about 10% of them, we showed this in 2003, about 10% of them carried a mutation in BRCA1 or BRCA2, which are one of the 3 very well-known mutations that are shared by this population. But then there were another few percent, another about 4% in total, so about 25% of all persons with all women with mutations that had mutation in one of the other genes. A couple of these are also known to be Ashkenazi-specific alleles. This one in CHEK2, which increases risk not nearly as much as BRCA1 and BRCA2, but it's not nothing. It's about 2 to 3-fold increased risk. Then other BRCA1 and 2 mutations that are private, and so on. So what this says to me is that if a woman has been tested for BRCA1 and BRCA2, she does not have a mutation in one of those genes, she has breast cancer, she has a family history, it's probably worth being tested now for the entire panel, even though that's not what one would necessarily do right at the beginning, given the enormous importance of these 3 mutations. The reason that I think it's so important that women in general in this country be tested is that in Israel, well, I shouldn't say in Israel, among Ashkenazi Jewish women, regardless of where they live, 40% of ovarian cancer is due to mutations in one of these genes. That's a huge amount of ovarian cancer, which we could make disappear if we knew who those women were before they developed the cancer. Second thing I want to show you is from my series of families of mixed ancestries in America. These are patients of all ancestries, mostly Caucasian, but mixed European ancestries, some Asian, some African American, who have patients of breast cancer who have, who have familial breast cancer but do not have a mutation in BRCA1 and BRCA2. And as you can see, there is a great mix of other genes that can be involved. Next result I want to show you is among women, again, of mixed ancestry. These are women who were ascertained in Seattle, so our Seattle population is mostly Northern European, who were presented with ovarian cancer and were not selected for either family history or age at diagnosis, and 18% of those women, as opposed to 40% of Ashkenazi Jewish women, but 18% of those women had a mutation that is responsible for their ovarian cancer. And note, please, that a third of these women with mutations were older than age 60 at diagnosis. Ovarian cancer is, of course, an older age at onset disease, so that's not remarkable, but it does suggest that all ovarian cancer patients should absolutely be screened because PARP inhibitors may be effective for many of these patients. It's the mutation that matters, it's not the age of the woman. So a 70-year-old woman who's diagnosed with ovarian cancer today in Tel Aviv, if she carries a BRCA1 or BRCA2 mutation, one should be thinking about PARP inhibitor, and one is. My young colleague, Barbara Norquist, who is a gynecologic oncologist who carried out the study I just showed you, also developed the odds ratios for each of the genes in that large DNA repair complex that I was telling you about previously. And as you see, for ovarian cancer, a number of genes have inherited mutations that play a substantial role in risk. Finally, very recently, there's been some interesting results apropos of advanced prostate cancer. So my young colleague, Colin Pritchard, who works in laboratory medicine at the University of Washington, set out to address the question of what about prostate cancer in mutation carriers. And the way that he and his colleagues did this was to ascertain, again through our center and collaborating with other centers, all men who had either been diagnosed with prostate cancer with a Gleason score of 8 or higher, or had had originally a prostate cancer with a lower Gleason score, but then had recurred with a prostate cancer with a Gleason score of 8 or higher, in other words, severe prostate cancer. And they sequenced them using the panel that, of course, I've told you about, and here's what they found, namely that 12% of these men, this is a mixed bag of ancestries, carried a mutation in one of the same set of genes. BRCA2 plays a bigger role here, probably not surprisingly, because we already knew it played a bigger role in male breast cancer, and BRCA1 also plays a role. Same genes with slightly different lineup in terms of their order of priority. So this raises the question immediately of what about PARP inhibitors for these men? So what are the themes that I have brought to bear? I think there are several of them. First, the mutations that are responsible for breast cancer, ovarian cancer, and prostate cancer in these genes are individually rare, but collectively fairly common. Second, that cancer develops when, but only when, we have an inherited and a somatic mutation in the same gene. Third, that the critical genes, when mutant, lead as their primary consequence to the loss of DNA repair. Then fourth, and I haven't discussed this, but somatic events in the same genes, that is a combination of two somatic events, two tissue-specific events, have the same effect. And finally, that from the point of view of the way we think about genetics, it's exactly what we've been hearing all morning up to now, that our story is a story of genetic heterogeneity, but not a story of polygenic effects. Many genes are involved, but each patient has an inherited mutation in only one gene. I'm going to skip several slides and come right to the punchline. Why is genetic testing important? We've talked about prevention, we've talked about treatment, and we've talked about families. And in consequence of all this, back a few years ago, I had the honor of winning a Lasker Award. And when you win a Lasker Award, it turns out, or at least it turned out at that time, they may have changed their policy, that the journal JAMA offers you to write a commentary. So I wrote a commentary with my friend Efrat Levi Lahad and her husband Amnon Lahad, who, as you know, works in family practice and primary care, and we made a proposition. And our proposition was that every woman regardless of her personal history, family history, everyone, be offered complete sequencing of BRCA1 and BRCA2 at about age 30 as a part of routine medical care, that it just be integrated with a visit to the gynecologist. That those women with a severe family history that is not resolved by BRCA1 or BRCA2 be referred for testing for all breast and ovarian cancer genes. And that women who have an unambiguously damaging mutation, the kinds I've shown you, be referred to a high-risk clinic for appropriate medical follow-up. Now, the reason I said the reason I made such a point about JAMA sort of being forced to offer me this opportunity because it was a policy, is that they posted above this commentary in bright red letters. The commentary below are the views of Dr. King and her co-authors and do not necessarily represent the views of the JAMA editorial board. I'm here to tell, it was 2014, I'm here to tell you that now, if I didn't show them that, they would claim they never wrote it. You know, now they say, oh, everyone knows this is true. We knew this all along. You know, let me tell you. The issues that were raised at that time, and they're now, that were undertaken very seriously for resolution in the intervening 4 years, were technical quality of the sequencing, how to interpret results, which is a big problem. There are many, many changes that don't have the effects that I've shown you. And it's been an important subspecialty of my field to develop both algorithms and experience with understanding what constitutes a change that matters versus a change that does not. Then yield. What proportion of women in a general population will carry a mutation that matters? In your population, the population of European Jews, whether in America or Israel, that fraction is 2.5%. We know that. What is it in, say, the American population generally, averaged across all ancestries? I think about 1%, based on just bits and pieces of evidence from various series. Cost is plummeting. Cost was a complete impediment previously. And then, of course, non-trivially, critically, follow-up for mutation carriers and follow-up for families. Those costs are enormous, but saving a woman's life is far more expensive than letting her die. So, to conclude, every ovarian or breast cancer patient with a BRCA1 or BRCA2 mutation that is detected after her diagnosis is a missed opportunity to have prevented that cancer. No woman with a mutation in BRCA1 or BRCA2 should die of breast or ovarian cancer. It is completely preventable, and it is absolutely unnecessary. Thanks very much.